This is CRISPR. You may have heard of it. Since its conception in 2012, CRISPR technology has become one of the biggest breakthroughs in gene editing by allowing scientists to use different enzymes to cut and edit genetic material within humans and other organisms. But this technology has a very interesting origin. A form of it was used in bacteria to defend against viral threats and attacks. And in light of the recent COVID-19 pandemic, some scientists at Sherlock Biosciences have come up with a technique that they aptly call Sherlock to help detect the presence of the SARS-CoV-2 virus using CRISPR technology. Let's break it down. So for this technology, the lucky enzyme is Cas13, which can cut and edit RNA, making it useful to fight RNA viruses. The next step is to identify a piece of the SARS-CoV-2's genetic makeup that is completely different from any genetic sequences within human genomes. Once we have that sequence, scientists will create a guide RNA that will help the Cas13 find the virus, along with reporters that light up once they're cut. Now that we have a blood sample, we can amplify the RNA within using a process called recombinase polymerase amplification, and then we can inject the reporters. Now it's time for the Cas13 to shine, and it starts looking for a match to its guide RNA. And as soon as it finds one, it starts cutting up everything inside of its path. This includes the reporters that were injected. If the reporters have not been cut, it means Cas13 never activated, and therefore the SARS-CoV-2 is not present. Now, scientists put this sample inside of what's called the flow detection system. Here, the reporters line up on different ends of the testing kit, depending on if they have been cut or if they have not been cut. This makes it very easy for scientists to spot signs of SARS-CoV-2 with just a glance. This test is much faster than PCR with a 30 to 60 minute completion time, making it one of the fastest tests you can find out there. It also requires little to no lab equipment since it's isothermal in nature. And finally, it is much more specific and sensitive than other tests, making it the best test for field use. And now you might be thinking, well, Ansh, if you can just detect the virus using the enzyme, and the enzyme is made to cut RNA, can't you just cut the virus? And you'd be right, and you'd be smart for thinking that, just like some researchers at Stanford University who have created a technique that does just that. It's called Pac-Man. While it still shows merit, it is currently in early stages of development and is nowhere near being ready for field use. Therefore, it might be a thing of the future. What intrigues me is the idea that we have used the help of bacteria to help us combat a different type of pathogen, viruses. The human mind is really beyond compare, but if not for the other organisms on this planet, we might not be so better off. I am Ansh Goyal, and I hope you learned something new today.